Hello friends, welcome to Basic Biology. Today we are going to discuss about the concept of adaptive immunity. In our last video, I discussed the basic concept of innate immune response, which is a general defense mechanism to prevent the entry of any microbes or pathogen inside our body. But in adaptive immunity, the interaction between the antigen and the immune component, say for example the antibody, is very specific. That is, antigens are recognized and eliminated from the body in a very specific way. Say for example, one medicine cannot treat many diseases. For each and every disease, there is a specific treatment. Similarly, in adaptive immune system, for each and every antigen, there is a specific kinds of interaction. So, this is called specific immunity. And this immunity has four major attributes. First one is the antigenic specificity. It means our immune system can recognize a specific antigen and it distinguish it from the another antigen. That is, our immune system has the potential to distinguish between two antigens, even they differ by a single amino acids. So it's a very unique property. Second is antigenic diversity. Now diversity means it's a recognition diversity. Our adaptive immune system has tremendous recognition diversity. That means it can recognize billion, trillions of sequences of antigen and react accordingly. Third one is the immunologic memory. Once the antigen is recognized by once the antigen has been recognized, the immune memory developed, and this is a very unique property of our adaptive immune defense mechanism, because during the second encounter with the same antigen, it induces the more heightened immune activity. That means within a very fraction of time, these antigens are eliminated from the body. But during the first exposure, it will take time. But during the second exposure, it will take a very minimum time to eradicate that antigen. So that is what is possible due to this immunologic memory. Third and fourth one is self, non-self recognition. Most of the body components are made by proteins, because we are nothing but the phenotype of proteins. Antigen are also proteins, but body can distinguish which are the self part of our body and which are the foreign part of our body. So this self and non-self recognition helps to identify the specific antigen or the foreign molecules. So the self and non-self recognition helps to recognize the foreign molecules or the foreign particles. The adaptive immune system is not an independent process. It doesn't mean innate is completely different part, adaptive is a different part. It doesn't mean like that. Because there are some phagocytic factors which play the crucial role during the activation of the non-specific or innate immune response. They also intimately playing some crucial role during the adaptive immune response. So both innate and adaptive immunity, they are linked with each other. Now we come to the different parts of adaptive immunity. Our adaptive immune system consists of two kinds of immunity. One is active immunity. In this immunity, the host or our body system directly interact with the antigen. That means when our own immune system activated, then it is called active immunity. Say for example, when we inject some vaccine, when we inject some hit kill germs inside your body, then our immunologic system becomes activated. So that mechanism is called active immunity. Whereas another one is the passive immunity. In this immunity, some immunologic products, say for example monoclonal antibody or some immunocompetent cells are injected into our body. That means our body's immune system is not directly activated, but the product of these immune systems are used to treat the disease. Recently in COVID-19, many people were affected and also cured. If scientists can utilize their antibodies to treat the other patients, so that is also called passive immunity. So in this way we can say this active and passive response together constitute our body's defense mechanism. An effective adaptive immune response is mediated by two kinds of immunity. One is CMI, that is cell mediated immunity, and another one is AMI, that is antibody mediated immunity. It is also known as humoral immunity. Now, cell mediated immunity is mediated by T lymphocytes, and humoral immunity is mediated by B lymphocyte. T lymphocyte or T cell can destroy antigen or any microorganisms without the intervention of antibody. But the condition is this antigen or the microorganism has to be presented by the membrane bound receptor, which is known as MHC, major histocompatibility complex, that I will discuss later on. So this T cell originate from the bone marrow. So if we see the diagram, in bone marrow there are different type of stem cells and they are known as hemopoietic stem cells. This hemopoietic stem cell undergoes differentiation to form two kinds of cell. One is known as precursor T cell, another is known as precursor B cell. This precursor T cell undergoes series of changes and passes through liver, then spleen, and where it processes to form a specific type of cell which is known as dependent lymphocyte, that is thymus dependent lymphocytes, and enter into thymus where it matures to form lymphoblast. And these lymphoblast, when stimulated by specific type of antigen, it can differentiate into three types of cells. One is T helper cell, and in each kind of T cell there is a specificity that it consists of a membrane bound receptor which is known as TCR, and along with this TCR there is a membrane bound protein which is known as CD4. What is the function of CD4? That I will discuss later on. Another one is TC or cytotoxic T cell. This is T helper, this is TC. It is also known as killer T cell, and it has a membrane bound protein which is known as CD8. Another type of cell is also present which is very few in number or less known about this cell, which is T suppressor cell. Now this T helper cell or TC cell can generate adaptive immune response via CMI, cell mediated immunity. T helper cell or TC cell is unable to recognize the antigen directly unless it is presented by any membrane bound protein or the membrane bound receptor, which is known as MHC. This MHC means major histocompatibility compatibility complex. There are two types of MHC. One is class 1 and another is class 2. Class 1 is recognized by CD8. That means it recognizes TC cell. And class 2 recognized by CD4. That is, it recognizes the T helper cell. And MHC is a membrane-bound receptor present in those cells which can represent the antigen via this receptor. And these kinds of cells are known as APC. That means antigen presenting cell. This cell can represent antigen using the MHC. That means MHC is present in APC. For an example, macrophage is a APC because it can phagocytose the antigen and it can represent the antigen using this MHC. So T helper cell can recognize these class 2 molecules. T helper cell can recognize antigen via these class 2 molecules and become activated and secrete some cytokines. Whereas in case of precursor B cell, now this precursor B cell originate in bone marrow and mature in bone marrow also. And once it is matured within the bone marrow, the naive B cell released from the bone marrow into the blood circulation. And this B cell, this naive B cell has a unique property. It has a membrane bound receptor which is an antibody molecule. And this membrane bound receptor is known as BCR, B cell receptor. And this is similar in structure with the antibody molecules. Now this B cell once encounter with the new antigen differentiated into two kinds of cell. One is effector B cell and another is memory B cell. Immunologic memory is a unique property of the adaptive immune response. So here this memory B cell is used for the future purpose, future encounter with the same antigen. This effector B cell is converted to plasma cell and these plasma cells secrete antibody which interact with the antigen to precipitate it or inactivate it. But one thing you have to remember that AMI or antibody mediated immunity is not an independent process because the secretion of antibody depends on the CMI, that is the cell mediated immunity. Because this effector B cell is converted to plasma cell is possible when the cytokines secreted from the T helper cells activate the effector cells. So that's why we can say the AMI is dependent on CMI, but CMI is not dependent on antibody. So this is the basic concept of immune system. In our later videos, I will discuss about the processing and presentation of MHC and the B cell T cell cooperations. So please follow us, and if you have any queries or questions, mail us, and definitely you can call me or WhatsApp me.